Hi guys, let's talk about flow cytometry and the FACS, or fluorescent-activated cell sorting. So what is flow cytometry? Flow cytometry is an analytical cell biology technique used to detect, identify, and count specific cells, for example like immune cells, based on cells' physical characteristic or markers called antigen on the cell surface, or some substance within cells that are unique to that cell type, and then determine different parameters of living cells in the heterogeneous mixture. Different parameters measured by flow cytometry include cell counting, cell characteristic and function, protein engineering detections, cell surface receptor detection, biomarkers detection, and diagnostic of health disorders such as blood cancer. These are basic components of flow cytometry. First, we have light source, flow chamber, optical system, light detector, and a computer or analysis workstation. In flow cytometry, different types of light scattering patterns and fluorescent emitting patterns involved in analysis of cell types. They include forward scattered and side scattered light, fluorescent emission, and multiparametric analysis. Forward scattered light refracts from the cells, counting along the same pathway through which it originally travels, and it helps to detect the size of the cells. Side scattered light refracts from the cells in a direction that is outside of the original light path, so it determines the granularity and the complexity of different cells. Additionally, fluorescent molecules of cells emit fluorescent light, helps the identification of different structure of the cells. Besides that, fluorescent dye or fluorescent tagged antibodies can also be used to label specific structure of the cells. Next, let's talk about how we do flow experiment by flow cytometry in the lab. The first step is sample preparation. A sample of cells is suspended in a fluid. Treat samples with specific dye to further define cell subtype, or stain cells with antibodies that bind to particular cells or key component of the cells. Then we run samples by flow cytometry. The fluid in which the cells are suspended passes through the very narrow channel so that cells are organized in a single file as they pass through the detectors. Next is signal detection. Flow cytometer. It contains one or more laser and a series of photodetectors that are able to identify certain characteristics unique to various cell types. The single cell suspension creates unique light scattering events that occurs when each cell passes through the laser light. These initial events are characteristic of the size and the shape of the cells, as well as the intensity of the signal that is generated by the specific dyes, thus creating patterns that reflect the cell type. The signals from the detectors are amplified and sent to a computer. They are converted to digital readouts displayed on a computer screen. So the last step is export data and the data analysis. We export raw data from the computer that is connected to flow cytometry and analyze it in the software like FlowJo. Next, let's talk about FACS, or fluorescent-activated cell sorting. FACS is a modified type of flow cytometry. The unique feature of FACS is its ability to physically separate each cell type in a heterogeneous mixture. It uses fluorescent-labeled target-specific antibodies to identify a particular cell type in the mixture. So FACS separates the heterogeneous cell mixture into two or more cell types in different containers. And FACS data is also analyzed in software like FlowJo. Don't forget to let me know if you have any other question in the comment section below. Bye.